Dear students, welcome to EPG Pathshala. I am Dr. Neetu Bhattacharya from Department of Zoology, Dayal Singh College, University of Delhi, Delhi. In this module, I will be talking about Giardia Morphology and Giardia Biology. After studying this module, you shall be able to know about the anaerobic flagellated protozoan parasites. You will be able to learn about the taxonomical classification of Giardia. You will be easily able to identify different morphological life cycle stages of Giardia, and you shall be able to evaluate complex microtubule cytoskeleton of the parasitic protist. And you can also analyze Giardia genomics resource database. Giardia is a microscopic parasite that is responsible for the diarrheal illness called as giardiasis throughout the world. It is the most common cause of waterborne outbreaks of diarrhea across the globe. These typical eukaryotic organisms are anaerobic flagellated protozoan parasites of the phylum Sarcomastigophora that colonize and replicate in the small intestine of several vertebrates. Giardia is also known as Giardia intestinalis, Giardia lamblia or Giardia duodenalis. Giardia lamblia is an important environmentally transmitted and an amitochondriate protist. It is a serious human pathogen that contributes to enormous worldwide burden of human diarrheal disease. No virulence factor has been identified till date. The Giardia lamblia genome comprises of 5 chromosomes with only 12 million base pairs distributed on it. This microscopic parasite is generally found outside or in soil, food or water which has been contaminated by infected human or animal feces. The parasite can spread by various ways of transmission. The most common of them is via water, such as drinking water and recreational water. Anything that comes into contact with the Giardia infected feces can become contaminated with the parasite. People become infected when they ingest the parasite. If we see the structure of the parasite, you will find that it comprises of an outer protective shell that allows it to survive outside the host body for longer period of time, and it also makes the parasite tolerant to chlorine disinfection. It is commonly found in soil, food or water that has been contaminated with the infected humans or animals' feces. The moment human being or an animal get infected with the Giardia, it starts residing in its intestine and is only passed out through its feces. Outside the host body, Giardia can survive for weeks or months. Giardia lamblia, which is a ubiquitous intestinal pathogen of mammals, is one of the members of the earliest diverging eukaryotic lineages. The life cycle of Giardia is uncomplicated as the environmentally robust cysts of the parasite present in the human feces are transmitted to a new human host mainly by waterborne transmission. Giardia lamblia has a distinct nucleus and nuclear membrane, well-developed cytoskeleton and endomembrane system, but it lacks other eukaryotes-like organelles such as nucleoli, mitochondria, peroxisomes and any of the component of oxidative phosphorylation. Let's come to the historical background of the parasite. The first recorded observation of Giardia trophozoite stages is usually credited to scientist Anton van Leeuwenhoek around the year 1681. Later on, in 1859, the protist was described in details by another scientist, Willem Lambel, who wrongly considered the organism in the genus Sarcomonas and named it Sarcomonas intestinalis. Almost 2 centuries later, there were two researchers, Grassi and Perancito, who started working on Giardia in between the year 1879 and 1888, and they only documented the acceptable morphological description of Giardia cyst and their links with the flagellated forms. The names Giardia lamblia and Giardia enterica were projected by two scientists, Kofoid and Christensen, in the year 1915 and 1920 respectively. The genus is also named after a French zoologist and applied entomologist, Alfred Matthew Giard. The species name Lamblia was extensively used during 1970s, but in 1980s scientists started using the name Duodenalis, and later on in 1990s other researchers promoted the species name Intestinalis. Now let's have a look on the taxonomy of the parasite. Giardia belongs to domain Eukaryota. Member of this domain possess two nuclei and a nuclear membrane. It belongs to the kingdom Protista. Member of this kingdom are eukaryotic and are generally unicellular organism, although few multicellular organisms also fall into this kingdom. The major key feature of Protista is that they are aquatic environment organisms. Giardia belong to phylum Sarcomastigophora. These are flagellated organisms and they are typically 10 to 20 micrometer in size. These include either free living or parasitic protozoan. Giardia comes under the class zooflagellates or Zoomastigophora, which means whip-bearing animals. The main feature of this class is how organisms use their flagella, one or more, for its movement. It falls under the order Diplomondida. Mostly parasitic organisms are there in this order. These organisms lack mitochondria or Golgi apparatus, but they have mitosomes. A mitosome is an organelle located in few unicellular eukaryotic organisms, and localization of iron sulfur cluster assembly proteins have been reported in them. Giardia belongs to family Hexamytridae. Members of this family contain 6 to 8 flagella, 2 true nuclei, and they are bilaterally symmetrical. The genus is Giardia. The genus Giardia include anaerobic parasites that feed and replicate in the small intestine of infected human beings and other mammals. Currently, 5 to 6 morphological distinct species of Giardia are recognized. These include Giardia murris in rodents, birds and reptiles, Giardia intestinalis or Giardia duodenalis and Giardia lamblia in mammals, which include humans, rodents and reptiles, Giardia agilis in amphibians, Giardia rd in the great blue heron and straw-necked ibis, Giardia cetacea in the bird garager, and Giardia microti in voles and muskrats. Giardia microti is the newly discovered species. Now let's compare the morphological and molecular features of different species of Giardia. Giardia agilis has long and slender teardrop-like median structure. The molecular data is not available for this species. Giardia lamblia is pear-shaped. It has 1 or 2 transverse and claw-shaped median body. It contains multiple genotypes. Giardia murris has short and rounded median body. Molecularly, it is distinct from Giardia lamblia. Giardia cetacea, rd and microti have same features as Giardia lamblia. Giardia rd and microti are related to the genotype of Giardia lamblia. Now, we will look into the morphological stages of Giardia. Giardia species have two major stages in their life cycle, the cyst stage and the trophozoite stage. Cysts are the resistant forms in which parasite is protected by an outer shell and are accountable for transmission of parasite that causes disease giardiasis. The cyst is relatively inert, allowing prolonged survival in various environmental conditions, while the trophozoite is the vegetative form and replicates in the small intestine, where it causes symptoms of diarrhea and malabsorption. Both cyst and trophozoites forms known as diagnostic and indicative stages that can be traced in the contaminated feces. Cysts of Giardia occur in the aquatic environment all over the world, and their population density is related to the level of fecal pollution or water pollution, mainly done by humans. After exposure to the host acidic environment of the stomach, cysts exist into trophozoites in the proximal small intestine. Infection of the host is initiated when the cyst is ingested with contaminated water or food or through direct fecal or oral contact. In new host duodenum, the trophozoite stages emerge from the infective cyst, which undergoes a mitotic division. Thus, through excistation, each cyst produces 2 trophozoites. Each of the 2 trophozoites remain attached to the epithelial cells with the help of adhesive disc and start feeding on the epithelial cells. In the lumen of the proximal small intestine, trophozoites multiply and remain as free or attached form by a ventral sucking disc to the mucosa. The trophozoites undergo rapid division. It divides within 72 hours by mitosis in the intestinal lumen. In diarrhea condition, many of the trophozoites get detached from the epithelial cells and excreted out with the intestinal contents, but they do not survive extensively outside the host body. However, encystation process occurs where trophozoites get transformed into cyst stage as the parasite transit toward the colon and excretes with the feces. These cyst forms can survive for long in the external environment and causes person to person transmission of the infective parasite. Let's come to the structure of the trophozoite. Now, trophozoites are bilaterally symmetrical and bear 2 nuclei without nucleoli that are positioned anteriorly. The trophozoites also have 2 median bodies and 4 pairs of flagella, namely anterior flagella and posterior flagella, caudal flagella and ventral flagella. Cytoplasm contain lysosomal vacuoles, ribosomal and glycogen granules. They also contain Golgi complexes that are noticeable in encysting trophozoites as stacked membranes indicative of Golgi complexes. The cyst is egg-shaped. It is 8 to 14 micrometer long and 7 to 10 micrometer broad in size. Each of the ingested cysts give rise to 2 trophozoites during excistation. Single cyst comprises of 4 nuclei, 4 median bodies and 8 pair of flagella, but the organelles arrangement pattern is not clear. Cysts are covered by 2 membranes, an outer filamentous and an inner membrane having 2 membranes. Complex microtubule cytoskeleton of this parasitic protist is vital for its motility, attachment to the host surface, cellular multiplication, intracellular transport and transitioning between its 2 morphological stages, that is the cyst and the trophozoite. The microtubule cytoskeleton is made up of both unique elements such as median body, ventral disc, funis and exoneme associated elements, and general structures of flagellated protists, like it contains 8 flagella and 2 mitotic spindles. Now, let us discuss the life cycle of the parasite Giardia. Cysts are resistant forms and are responsible for transmission of giardiasis. Both cysts and trophozoites can be found in the feces as diagnostic stages. The cysts are generally hardy and can survive several months in cold water. Infection occurs by the ingestion of cysts in contaminated water, food or by the fecal oral route. In the small intestine, excistation releases trophozoites. Each cyst produces 2 trophozoites. Trophozoites then multiply by longitudinal binary fission, remaining in the lumen of the proximal small bowel, where they can be free or attached to the mucosa by a ventral sucking disc. Encystation occurs at as the parasite transit towards the colon. The cyst is the stage found most commonly in non-diarrheal feces. Because the cysts are infectious, when passed in the stool or shortly afterward, person to person transmission is possible. Giardia can be transmitted by various modes like swallowing Giardia directly from contaminated surfaces, for example bathroom handles, changing tables, diapers or toys that contain feces from an Giardia infected person or animal. Drinking water procured from infected water sources such as untreated or improperly treated water from lakes, streams or wells. Swallowing recreational water, especially in lakes, rivers, springs, ponds and streams, while swimming or playing in Giardia infected water. Eating undercooked food that may contain Giardia parasite, or traveling to regions or countries where giardiasis is common. There are intestinal symptoms also. Stool samples from the patients are generally examined for the detection of the motile trophozoite and cyst of Giardia. According to Centers for Disease Control and Prevention, USA, Giardia infection can cause a variety of intestinal symptoms, which include diarrhea, gas or flatulence, greasy stool that can float in water, stomach or abdominal cramps, upset stomach or nausea, dehydration, weight loss. But few people with Giardia infection shows no symptoms at all. There are several complications which are actually associated with Giardia infection, like dehydration. As a result of severe diarrhea, dehydration occurs in the body, due to which not enough water is present within the body to carry out the normal body functions. Failure to thrive. Chronic diarrhea from giardiasis can lead to malnutrition and retard infants or children physical and mental development. Lactose intolerance is another complication. Giardia infected person suffers from lactose intolerance, that is the inability to properly digest milk sugar lactose. The problem may persist long after the giardiasis infection has cleared. There are several prevention measures to control infection with Giardia parasites. These include practice of proper hygiene and sanitation. We should avoid drinking water such as recreational water that may be contaminated. we should avoid undercooked food, and we should also avoid contact and contamination with the human and animal feces. There are several common medicines available in market to control giardiasis. The common one is metronidazole. It is the most commonly prescribed antibiotic for giardia infection. Side effects may include nausea and a kind of metallic taste in the mouth. Tinidazole is another antibiotic available in the market with the common name Tindamax. It has many of the same side effects like metronidazole, but it can be given in a single dose. Nitazoxanide is available for children. It comes in a liquid form and is easier for children to swallow. Side effects may include nausea, flatulence, yellow eyes and brightly colored yellow urine. Let's talk about the Giardia Genome project. Giardia lamblia genome has 12 million base pairs spread over total 5 chromosomes. The genome carries eukaryotic features such as linear chromosomes with telomeres. The genome contains multiple tandem repeats of pentamer TAGGG, which is similar to other eukaryotic organisms. The haploid genomic size of Giardia lamblia is approximately 11.7 MB, and it was first reported by Morrison et al. In 2007. Giardia lamblia has 5012 protein coding genes and also has all 4 of the core histone proteins. No virulence factors has been recognized in this genome till date. Giardia DB is the Giardia database. Giardia database is a member of pathogen databases that are housed under the NIAID funded UPathDB Bioinformatics Resource Center umbrella. The UPathDB team is mainly distributed between the University of Pennsylvania and the University of Georgia. The official website of Giardia database is www.giardiadb.org/giardiadb/. The Giardia Genome project, that is Giardia DB analysis, helps us to provide an insights about the origin of nuclear genome organization, the various metabolic pathways used by parasitic protists, and the cellular biology of host interaction and mechanisms used to avoid host immune systems. The Giardia DB1 version was released on 1st October 2007 and updated versions are now available. Giardia DB 1.1 release contains the genome and annotation information for the Giardia lamblia ATCC 50803 and WBC6 isolate and was published in Science Journal in 2007. The new data available includes all genes query, 10 SAGE tag libraries, microarray data and proteomics data from trophozoites and quantic coordinates. Giardia DB of UPathDB family of functional genomic databases contains the genome database for Giardia lamblia. It uses the same bioinformatics framework as other UPathDB sites supporting fully integrated and searchable databases. Genomic scale data available via these integrated genomic resources may be queried based on BLAST searches, annotation keywords and gene ID searches, geoterms, sequence motifs and other protein characteristics. Functional queries may also be formulated based on transcript and protein expression data from a variety of platforms. Further, phylogenetic relationships may also be searched. The ability to combine the results from independent queries and to store queries and query results for future use facilitates complex genome-wide mining of functional genomic data. Now, let us summarize the module. Giardia lamblia, also called as Giardia intestinalis or Giardia duodenalis, is a flagellated unicellular eukaryotic parasite, commonly causes waterborne outbreaks of diarrhea and occasionally foodborne diarrhea globally. The diarrhea disease caused by the Giardia species is known as giardiasis. Giardia lamblia have two main stages in the life cycle, the cyst and the trophozoite. Infection in the mammalian host such as humans is caused by cyst ingestion via contaminated food, water or direct fecal oral contamination. Giardia database is available online. Whole genome wide data tools helps us to reveal phylogenetic associations and evolutionary history of these microbial pathogens. Thank you.